Okay, so uh, the intestinal juice contains so these almost completed digestion is happening by the pancreatic enzymes, after which they will be entering into the intestinal mucosal cells or the enterocytes, where they encounters with the intestinal enzymes, that is intestinal enzymes present within the intestinal juice. So when this food enters into the intestinal or the enterocytes, what happens? The enterocytes releases intestinal juice, and this intestinal juice contains enzymes which are responsible for causing the final digestion of the simpler peptides. Okay, so like that of in the carbohydrate digestion, once the digestion is almost over, when they enter into the enterocytes, what are happening? We'll be having only disaccharides, trisaccharides, or monosaccharides itself. So monosaccharides cannot be finally again we cannot break down further. So it will be remaining as such, like glucose, fructose, galactose, etc. While we'll be having sucrose, sucrose will be there, lactose will be there, right? Maltose will be there. So how are the digestion of those dipeptides and maltotriose will be there? How are they carried out? They're carried out by the intestinal enzymes, or the enzymes represented in the intestinal juice. Like we'll be having disaccharadases like sucrase, lactase, maltase, etc. So similarly, in the case of proteins also, once the digestion is carried out by the pancreatic enzymes like trypsin, chymotrypsin, elastase, and the carboxypeptidases, will be having almost simpler peptides, that will be having either dipeptides or simpler peptides like tripeptides, etc. Right? That is almost the breakage. The degradation will be over by the pancreatic or carried out by the pancreatic enzymes. Now these simpler molecules enters into the enterocytes. Again, it is carried out with the help of some transport mechanism that we'll discuss. Again, in the enterocytes or in the intestinal luminal cells, what is happening? The intestinal juice carries out the complete digestion of the peptides to give rise to individual amino acids, right? So dipeptides, tripeptides, amino acids itself will be remaining there, and they are carried out by the digestion is carried out by the intestinal enzymes, and there are three different intestinal enzymes present in the intestinal juice. One is the entropeptidase, whose function is to carry out the activation of trypsinogen, few molecules of trypsinogen, that is in order to carry in the presence of a pH of 5.2 to 5.2 to 6. Okay, so that is the function of entropeptidase, which is present in the intestinal juice. The second enzyme present is the aminopeptidase. So it's again, as the name indicates, it's an exopeptidase. That is, this peptidase enzyme can act on the N-terminal end, so that individual single amino acids will be released at a time, right? So that is an aminopeptidases. Now, these aminopeptidases cannot carry out the digestion of dipeptides and all. So the dipeptides, if a molecule is cleaved into dipeptide forms, that is two amino acids joined together by a peptide form, they cannot be further processed by this aminopeptidases and all. So that is carried out by enzymes known as dipeptidases. So that again these dipeptides will be cleaved into individual amino acids. With this, the digestion will be over. Right? Now they are entering into the absorptive stage. So now all the enzymes, all the amino acids, all the protein molecules are having complete digestion. They are all split up into individual amino acids, and they are now ready for absorption. So this is a all-round or the flow chart of a digestion of proteins. So in the mouth, there is no enzymes capable of carrying out the digestion. So in the mouth, there is nothing happening for the protein part of the dietary sources. On entering into the stomach, pepsin and it is not renin, it is renin. The spelling is wrong. It is R-E-N-N-I-N. Renin is seen in the kidneys, glomerulus, right? So this is renin. Okay, so renin and pepsin. Renin is acting in the newborns, while pepsin is acting in the adults, initiating the digestion. Curd, the milk will be digested to curd by pepsin and renin also. Then this partially digested food enters into the pancreas, where they are acted upon by the entopeptidases, trypsin, chymotrypsin, and elastase, while the digestion is initiated by the trypsin molecule. Again, trypsinase is present in the form of trypsinogen. The trypsinogen is converted to trypsin by means of the intestinal enzyme entropeptidase. So entropeptidase carries out the conversion of few molecules of trypsinogen to trypsin. This trypsin carries out the further activation of the remaining trypsinogen molecules to trypsin. Also carries out the activation of chymotrypsinogen to chymotrypsin, proelastase to elastase, and procarboxypeptidases to carboxypeptidase. So all out of these different enzymes present in the pancreatic juice. These three are endopeptidases, that they are deep within the molecules. They can break the peptide bonds deep within the molecules, while carboxypeptidase acts on the C-terminal end. While carboxypeptidase acts on aromatic, the aromatic amino acids or the aliphatic amino acids with branch side chains on the C-terminal end, while carboxypeptidase B will be cleaving the peptide C-terminal end with basic amino acids. Okay, now these almost digested product enters into the intestine, where they are acted upon by another exopeptidase, the aminopeptidase. A typical example is leucine aminopeptidase. So leucine aminopeptidase carries out the digestion of leucine. Okay, so if the aminopeptidase, if the peptide bond is having leucine there, if the N terminal is having leucine there, then it is acted upon by leucine aminopeptidase. So each amino acid has got an aminopeptidase also. A typical example is the LAP, leucine aminopeptidase. And these aminopeptidases cannot cleave the dipeptides. So the further digestion, if there is any dipeptides remaining, then they are cleaved by dipeptidases to give rise to individual amino acids. Digestion in the mouth. So the mouth is only having the function to carry out the lubrication of the food, right? So after mastication and chewing, they will be entering into the gastric cavity, where it is carried out by HCl. HCl moon functions in there to maintain the pH, to carry out the denaturation and all, right? Then pepsinogen to pepsin conversion, right? And renin is present in the infants. Then stomach secretes a strong HCl, converting pepsinogen, proenzyme pepsinogen to pepsin. It also carries out another important function, that is, they denatures the proteins, they maintains the reduced pH, activates pepsinogen. They can kill bacteria. HCl can kill the bacteria and enables the vitamin B12 absorption also. These are another functions carried out by the HCl, right? So the denaturation of the proteins, maintaining the low pH or the acidic pH, conversion of pepsinogen to pepsin. Fourth function is killing of the bacteria, and fifth function is absorption of vitamin B12, cobalamin. Right now, pepsinogen, what it is carried out by the secretion is carried out by the chief cells of the gastric parietal cells, carries out conversion of pepsinogen to pepsin by HCl. So now, once small quantities of pepsin is formed, they undergoes autocatalysis to give rise to more and more pepsinogen will be converted into pepsin, like that of trypsin. Pepsin carries out the digestion of peptide bonds where there meets the aromatic amino acids and methionine. They converts proteins to proteases and peptones. Can also carry out the curdling of milk. So hence it is also known as chymosin. This is a renin, right? It converts casein into paracasein and calcium paracaseinates. Right now, after this, what happens? They enters into the duodenum, where they enter and the encounters with cholecystokinin, pancreasamine, etc. The endopeptidases will be there. The exopeptidases are C-terminal and carboxypeptidase and aminopeptidase. Pancreatic peptidase proenzymes, trypsin, chymotrypsin, elastin, carboxypeptidase A and B. Then trypsin, enterokinase acted upon by trypsinogen to give trypsin, chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase. Chymotrypsin, these are all discussed. Carboxypeptidase, they are the zinc containing enzyme. It's very important. Okay. So this is another chart. Like pepsinogen can be converted into pepsin, trypsinogen to trypsin. This trypsin ആണ് ബാക്കി എല്ലാവരെയും activate ചെയ്യുക. So trypsin. So this is the overview of digestion. What is happening? This is the N-terminal end, this is the C-terminal end. So carboxypeptidase always acts on the C-terminal end. And again, if the C-terminal end is having basic amino acids, it is acted upon by carboxypeptidase B. Or if it is acted upon, if it's having aromatic amino acids or valine leucine isoleucine, then it's acted upon by carboxypeptidase A. Now, absorption. So now we are having the peptides broken up to give rise to individual amino acids with the help of various types of proteolytic enzymes, which includes both the enteropeptidases and exopeptidases. Now, how is absorption happening? The absorption of amino acid is an energy requiring process. So it requires the consumption of, or in presence of ATP, with the help of transport protein, the amino acids are taken into the cells, intestinal cells. So the transporter is sodium amino acid co-transport. That is, along with an amino acid, sodium ion also will be entering into the cells. So it's an energy requiring process and it's an uniport system. That is, both sodium and amino acids are moving in or entering into the intestinal epithelial cells, crossing the brush borders. They are moving in the same, or they are traveling in the same direction. The transport mechanism which enables the absorption of the amino acids are known as sodium amino acid co-transport mechanism, which is an energy requiring process. So what is happening? This is a brush border of the intestinal lumen. So now we are having the individual amino acids and the sodium ions, right? So with the help of the sodium potassium pump, what is happening? First, the concentration of the, like that of our sodium glucose transport, right? In the case of carbohydrates also there was a mechanism, right? Sodium glucose transport mechanism. The same way, sodium pump. So in here also sodium pump will be there. Sodium pump will be releasing the various amino acids and sodium first out of the intestinal lumen, so that the concentration is reduced. Then only these molecules can enter into the intestinal epithelial cells, right? So this other sodium ions, suppose these red colors molecules are the sodium ions, and this blue, yellow and lilac colored are the amino acids. So what is happening? First, we know that sodium is an extracellular ion. So if sodium is entering from the intestinal cell lumen into the intestinal epithelial cells, what has to be first done? First, the sodium ions in the epithelial cells has to be taken out. Okay, and that is carried out with the help of sodium potassium pump. So that sodium will be coming out while potassium will be entering. So these triangular shaped molecules are the potassium ions, while the red colored things are the sodium ions. So we know that the sodium pump acts in such a way that in a single transport, 3 sodium ions will be taken out, while 2 potassium ions will be taken in. Okay, so the first event to be taken place is the removal of extra sodium ions from the intestinal epithelial cells. Then only the sodium which is now obtained from the diet can enter into the epithelial cells. Okay, so the first thing is to be the functioning of the sodium pump, right? And this is an energy requiring process. This is not an energy requiring process. This functioning of the sodium pump is an energy requiring process. So once the sodium ions are out into the blood, into the systemic, into the portal circulation, that is, now these ions enters into the blood and they will be taken into the liver. Now the concentration of sodium ions will be reduced. As a result of which, what happens? The dietary sodium can easily enter into the epithelial cells, right? And that is carried out with the help of sodium amino acid transport. So this is a transport protein which is present on the surface of the brush border, right? So this is our lumen, and in the lumen we are having the simplest amino acids, or the proteins are now converted to their simplest form, that is amino acids, and also sodium ions are there. So these one sodium ion will be binding to its sodium binding site, and the amino acid will be binding to the amino acid binding site, and the protein now faces to the cell interior and will be releasing the amino acid followed by sodium, and again it will be facing back, so that the next sodium and the next amino acid come again, bind to their corresponding sites, and they are taken into the cell. So this is how the co-absorption is happening, right? So transport system, small and neutral amino acids, large neutral amino acids, basic amino acids, acidic amino acids, and amino acids, and glycine. This is glycine is something different. Otherwise, the amino acids transported. All these amino acids will be released or absorbed with the help of the sodium amino acid co-transport mechanism, except for the case of basic amino acids and all, right? So this is how the absorption is taking place in the body. The L amino acids, the condition again is that all the amino acids should be L amino acids, while the carbohydrates, the sugar should be D-glucose, D-fructose ethanol. So the L amino acids are absorbed by an active carrier-mediated mechanism. Why it is an active process? Because first the sodium pump has to take extra sodium ions from the epithelial cells. It has to release out the extra sodium ions from the epithelial cells into the blood. Then only the diet-derived sodium can enter into the epithelium. They can undergo absorption. So the rate of absorption of L amino acid is independent of the concentration gradient, but sodium. So how can the the amino acid, the intestinal concentration of the L amino acid will be reduced.